Okay, now let's talk about the neurotransmitters. Okay, I told you that in a chemical synapse we have release of the neurotransmitter onto the postsynaptic membrane. So these neurotransmitters can be either small molecule or large molecule neurotransmitters. So two categories include small molecule neurotransmitters and large molecule neurotransmitters. Large molecule neurotransmitters are also called neuropeptides. Okay? So there are four classes of small molecule neurotransmitters. Class 1 is acetylcholine. Class 2 includes some amines, like norepinephrine, epinephrine, dopamine, serotonin, histamine. All of them have a amine group inside this chemical structure. Right? So amines, actually some people say amines, so it's not correct, but I think amines is better. Okay? Amino acids, like GABA, glycine, glutamate acid. Class 4 is nitric oxide, which is a gas. Okay? So four classes of small molecule neurotransmitters include acetylcholine, amines, amino acids, gases. In the fourth class, we have nitric oxide, carbon monoxide, and other gases. Okay? But they are different from other categories. So in a chemical synapse, we have vesicles that contain the neurotransmitter. But gases cannot be confined in a vesicle, they go out. So we don't have vesicles for storing gases. Okay? Whenever they are needed, they are made at the same time. Okay? So, they are rapidly acting neurotransmitters, these categories. And they are used mainly for acute responses of the nervous system, like for sensory signals to the brain and motor signals to the muscles. So in these examples, or in these cases, we will have small molecule neurotransmitters. So try to memorize the categories. Okay? Large molecule neurotransmitters, also called neuropeptides, include some familiar proteins for you. For example, you know prolactin. It's a hormone, right? You know insulin, you know glucagon, you know angiotensin 2, you know gastrin, substance P. So all of these are some peptides, right? You know that hormone is secreted from a gland into the blood and goes through the blood to distant targets. Targets located far away from the gland. Right? So this is the action, and they act on the target. Okay? So this is the action of hormone. So one example is insulin, secreted from the beta cells of the pancreas. Right? And you know their action. For example, thank you so much. They, for example, change glucose level in the blood. Right? But here we are talking about neurotransmitter. Insulin is a hormone, but it's also a neurotransmitter. So the same chemical can be used as a hormone or as a neuropeptide or as a neurotransmitter. If it's secreted from a gland into the blood, it's a hormone. But if it's secreted or released from a presynaptic cell onto a postsynaptic cell, it's a neurotransmitter. Got it? So, you can see a long list of neuropeptides, but they are slowly acting neurotransmitters. So they act slowly, and the action is prolonged. So they can change the number of receptors, they can open or close ion channels for a long time, they can change the number of synapses, they can change the size of the synapses. So that's why sometimes we call them neuromodulators. Neuromodulators. So they modulate the action of a synapse. Okay, so imagine that you have a synapse here. Okay? So it releases a neurotransmitter like acetylcholine. It binds to the postsynaptic cell, postsynaptic neurotransmitter, right? Postsynaptic receptor. So maybe we also release one of these neuropeptides, like substance P. Okay? So it may act on some channels. Okay, so this is the standard action of a synapse, or the standard action of a neurotransmitter, right? But substance P can also increase the size of this synapse. Or it can increase the number of vesicles that are released, or the number of receptors, or the number of synapses. Okay? So they have some prolonged actions. So neuropeptides are the second category of neurotransmitters. So you know that some receptors, actually for some neurotransmitters we may have several receptors. Like an example, acetylcholine has nicotine receptor and muscarine receptor. You know that? Yes or no?
Yes.
Yes. So you know that in a skeletal muscle, acetylcholine binds to nicotinic receptor. So contraction of the muscle is the result of activation of nicotine receptor. But in the heart muscle, acetylcholine binds to muscarine receptor. So it decreases heart rate. Right? So in the skeletal muscle, acetylcholine is excitatory. In the heart muscle, acetylcholine is inhibitory. Why? It has two different receptors. One receptor is excitatory, another receptor is inhibitory.
We should remember all of this. Should remember all of this.
Peptides.
Yes. Yes. For example, I may give you a question like this. Which of the following is not a small molecule neurotransmitter? Dopamine, serotonin, nitric oxide, substance.
Substance.
In order to answer this question, you should be familiar with these. At least some highlighted, some important ones. So for example, you know that prolactin is a peptide. You're familiar with that.
Yes.
You know the insulin.
Insulin.
You know that, for example, calcium. You know vasopressin. You know oxytocin.
لازم ناخذها
So you should know only the concept. The concept is that all of these are peptides, but dopamine is not a peptide. Serotonin is not a peptide. Glutamate is not a peptide, it's an amino acid. Right? Which of the following has the slowest action? Dopamine, serotonin, nitric oxide, substance. Which one is? So let me ask this question 2 slides later. Okay? So, okay. After the release of the neurotransmitter, its action should be terminated. Because we don't need the action for a very long time or forever. So it should have a very fast action and a very short action. So we need termination of the action. Termination is actually made or happens by several mechanisms. One of them is the degradation of the neurotransmitter. So the neurotransmitter is broken down. Okay? So one mechanism. The second mechanism is diffusion. It can go out of the synapse. The third mechanism is reuptake. So the neurotransmitter is released, norepinephrine in this example. It binds to the postsynaptic receptor, after that, we don't need it. It goes back to the axon terminal. This is called reuptake. Reuptake is made by a transporter. So in this case, norepinephrine transporter. Norepinephrine is the neurotransmitter. Transporter is norepinephrine transporter. So it goes back into the cytoplasm, and it should reach the vesicle. Onto the vesicle, I mean in the membrane of the vesicle, we have another transporter, vesicular monoamine transporter. So this transporter moves neurotransmitter into the vesicle. So we have two different transporters for the reuptake of the norepinephrine. So in this way, the action of the norepinephrine is terminated. For some neurotransmitters, reuptake is the main process, is the main mechanism for termination of action. For other neurotransmitters, degradation is the main one. For still another type of or category of neurotransmitters, diffusion is the main mechanism. Okay?
How neurotransmitter travel through synaptic scala?
Again?
Neurotransmitter, how they travel through the synaptic scala?
How they travel? So actually, they travel in the
From
synaptic.
the pre to post synaptic scala.
Yeah, from here to here, diffusion.
Just diffusion, they
Yeah.
doesn't need anything.
No, they don't need anything. Because the cleft is very small. Up to 5 micrometer, right? Or nanometer. Nanometers. Right? So it's very small. 40 nanometers, remember.
Yeah.
20 to 40 nanometers. Yeah, 200 or 400 nanometers. So it's very small, so it diffuses from the presynaptic to the postsynaptic. So reuptake, remember this. Neuropeptides are made in the cell. Why? Because they are peptide. You know, peptides are made by ribosomes. Where are they? Rough endoplasmic reticulum. So endoplasmic reticulum is in the cell. And nucleus is also in the cell. Inside the nucleus we have the genes. So neuropeptides are made by mRNA. So we need mRNA. So it comes from the genome. Right? Then mRNA binds to the ribosomes. Ribosome translates the mRNA into protein. Protein is confined in the Golgi apparatus, right? In the plasmic reticulum, then Golgi apparatus. Then they are actually stored in vesicles. Inside the vesicle, we have splitting. You know splitting? So a long sequence of amino acid is cut from some places, right? So one peptide is converted into several smaller fragments. And this is necessary for activation of neuropeptides. This is splitting. So they are packed in vesicles. Then vesicles should go from the cell body to the axon terminal. From the cell body to axon terminal. This happens by way of axonal streaming. So they are released when action potential arrives. And it needs calcium. Okay, there are some other differences. So now, which of the following means axonal streaming? Dopamine, serotonin, nitric oxide, substance P. Got it? So I may ask about large or small molecule. I may ask about slow or fast action. I may ask about axonal streaming. I may ask about the place of synthesis, soma or axon terminal. Okay, so all of them are possible, right? So compare these two. So classic transmitters, peptide transmitters. Synthesis. Left terminal, cell. Activation. Classic transmitters, small molecules, are active after their synthesis. But peptides need splitting. They should be cut. Right? Vesicles. Small molecules are stored in small vesicles that are clear under the microscope. But neuropeptides are stored in large vesicles that are electron dense. Release. Standard transmitters are released in a synaptic cleft. But neuropeptides may be released from other sites of the neuron. They don't need to be released into the synaptic cleft. Sometimes they can be released outside the cleft. It's possible. Action of the neurotransmitters is terminated by reuptake, neuropeptide by proteolysis or diffusion. Latency of action, duration of action. Small molecule neurotransmitters have short latency, short duration. Neuropeptides have long latency and longer action.
Neuropeptide is just made by the ribosome which are attached to the endoplasmic reticulum or the free ribosome.
Neuropeptides, so they are made in endoplasmic reticulum. So first they enter endoplasmic reticulum, then Golgi apparatus, then they are packed in vesicles.
So the free ribosomes which are present in the cytoplasm, they
No.
can also make?
No. If they are made by free ribosome, then they cannot be packed into the vesicles.
Yeah. So there is any ribosome in dendroid and axon?
Dendrite and axon, we don't have. No. Actually, in large dendrites, we have some endoplasmic reticulum. Okay? So, for example, you can consider a neuron with soma and larger dendrites and then smaller dendrites. So the base of the dendrite, the tip of the dendrite. The base is larger. So inside the large part of the dendrite, we have some endoplasmic reticulum. Okay? So it's possible for neuropeptides to make here inside the dendrite. Okay. So, co-release, co-transmission. I told you that a vesicle contains neurotransmitter. In the past, we believe that in each vesicle, we only have one type of neurotransmitter. So, for example, if in a vesicle we have dopamine, we have only dopamine. We don't have any other type of neurotransmitter inside this vesicle. Or we also believe that a cell that makes, for example, dopamine doesn't make like serotonin. Okay? But now everything was changed actually. We know that in a vesicle, in a single vesicle, it's possible to have more than one type of neurotransmitter. Also, in a cell or in an axon terminal, it's possible to have a vesicle with, for example, one type of neurotransmitter, another vesicle with another type of neurotransmitter. Okay? So this is the basis for co-release and co-transmission. So in this example, you can see that in this vesicle we have two neurotransmitters, the purple one, the green one. So if action potential arrives and opens the calcium channel, calcium triggers release of neurotransmitter, exocytosis. By exocytosis, you will have release of both of these vesicles, both of these neurotransmitters. So we have co-release. Right? Co-release. So examples. co-release of GABA and glycine. So both GABA and glycine are in the same vesicle. Co-release of glutamate and dopamine in the ventral tegmental area. So for example, the purple one is glutamate, the green one is dopamine. It's possible to release both of them. So this is called co-release. In another type, we have co-transmission. So here you have some vesicles that contain neurotransmitter 1, another vesicle which contains neurotransmitter 2, right? But they are not always released at the same time. So here you can see that this type of neurotransmitter only needs one action potential. But the purple one needs several action potential. Why? Because exocytosis of these neurotransmitters depends on different concentration of calcium. This one is very sensitive to calcium. This one has lower sensitivity to calcium. So, one action potential causes influx of calcium. Only a small increase in calcium concentration is enough for release of this one. But it's not enough for release of this one. It needs more calcium. How we can have more calcium? By having several action potentials. So action potentials come one after the other one because repetitive influx of calcium concentration goes very high, and now it's enough for the release of this neurotransmitter. So if you have only one action potential, this type of neurotransmitter is released. If you have several action potential, then both of them are released. So this is called co-transmission with different calcium sensitivity. Another type of co-transmission is like this. One action terminal only contains one type of vesicle, the other one has another type of vesicle. When action potential arrives, it goes this way and this way. It causes release of this neurotransmitter onto one synapse, another type of neurotransmitter,
Another side.
another synapse. So, co-transmission with spatial segregation. Synapses are separated.
The first video, Mr. Please. No, video. Yeah. When we need like entering the first room and a huge amount for opening the second one. When we're entering like in the starting, we're entering a little bit. So the first one was sensitive to it. So in case of the second transmission, we need to open both of them. Like when we open the starting amount, it will open the first one because it's sensitive.
Yeah. So lower concentration of calcium is enough for release of this.
Yes, and we want a huge amount for this one.
Yeah.
So if we're entering a little bit, it will be open also.
Yeah.
So
So if we have several action potential,
yes.
then we have higher concentration of calcium. First, it releases this one, then it releases this one.
Yes.
But both of them are released. Right? If the calcium concentration is low, only
Just one.
this one is released.
Excuse me, sir. Is it possible for two inhibitory or excitatory neurotransmitters in the same vesicle?
Yes, it's possible. In the same vesicle, you mean like this or like this?
You mentioned the example
Both.
co-release of GABA and glycine in the spinal cord.
Yeah, co-release of GABA and glycine. Both of them are inhibitory.
Yeah.
Yeah. But glutamate and dopamine.
So
Glutamate is excitatory. Dopamine is sometimes inhibitory.
So they can.
So dopamine has several types of neurons receptors. Some of them are inhibitory, others are excitatory. So generally, we consider dopamine as an inhibitory neurotransmitter. Generally, okay, but it has also excitatory action. So it's possible to have an excitatory and an inhibitory neurotransmitter at the same time in the same vesicle.
Yeah, my question was the same, but glutamate and GABA is the same vesicle, they are open together.
Yeah, they open it. Sometimes we may have a small molecule and a large molecule neurotransmitter in the same vesicle. But generally, we have them like this. Small molecule in one type of vesicle, large molecule in another. So co-transmission usually happens for a small and large molecule neurotransmitter. Got it? So we have 10 slides more. Yeah, 10 slides more. And we have 15, 30 minutes.
15.
Okay. I will try my best. Okay. So,
Not asking.
neurotransmitter, small or large, is released by the receptor, metabotropic or ionotropic. Right? It opens some channels. This channel can be a sodium channel, a potassium channel, a calcium
Chloride.
channel, a chloride channel, everything.
Yes.
Right? So, let's imagine that we have a cell with sodium channels.
Okay.
Neurotransmitter opens the sodium channels. What happens next?
The sodium will come inside of the cell,
Sodium.
and the
Sodium goes
concentration will.
Inside or outside?
Sodium?
Yeah.
No, inside.
Inside. Why?
Because the concentration. Because the concentration will be small, so
Concentration of sodium is higher outside.
yeah, outside will come inside.
So it goes inside. What else?
The potassium will go outside.
No, I mean, so one
The concentration
reason for
of-
influx, one reason for
Yeah.
inward movement is concentration.
So the voltage will increase because the sodium is positive,
Yeah.
and
Inside the cell is negative.
and will be positive.
Exactly. So, sodium goes into the cell because of two reasons. One is higher concentration outside.
Yes.
The second one is that sodium is has positive charge. Inside the cell is negative, so negativity pulls the positivity.
Positive, yeah.
Right? So we have two driving forces: concentration, electrical gradient, electrical difference.
Mm.
Right? So, channel is open, sodium goes in.
Inside, yeah.
When this movement is ended, I mean, do we have inward movement of sodium as long as the channel is open? No. Why?
Because later the concentration of sodium in the cell increases, and then after that, it will not be like the difference in concentration won't be enough to push the sodium into the cell.
Yeah, theoretically this one is correct. But realistically, it
It
doesn't
has to
happen.
do with the membrane potential.
Membrane potential is
Because
very
the membrane, the potential inside the membrane increases, and then after that, there will be electrical stability, and there will not be.
Good. Usually the channel doesn't remain open enough for changing the concentration of the sodium inside the cell. So concentration always is higher outside. Okay?
Mm.
I mean, opening of the channel is not enough for changing the concentration. But only a small movement of sodium from out to in is enough for changing the negative potential inside the cell. Sodium is positive, when it goes into the cell becomes more positive. And this positivity actually is against the further movement of sodium. Now, how can we calculate the potential for stopping the movement of sodium?
To the nerve.
Stimulus.
Nernst potential. So Nernst potential is a potential of the membrane at which there is no further movement of sodium inside. Right? How can we calculate this? So, electromotive force for sodium equals -60 times logarithm of concentration inside to concentration outside. So -61 times 1, so 110, right? -10, so it becomes +61. So at this membrane potential, sodium doesn't go in the cell. So in the resting state, membrane potential is -70 millivolts. Sodium channels are opened. Then sodium goes in. This movement increases the positivity. When it reaches +61 millivolt, sodium doesn't go into the cell anymore. It stops. We call this Nernst potential for sodium. We can calculate Nernst potential for potassium. If we calculate it, it's about -87. So, a neurotransmitter is released, opens potassium channels. Potassium goes out. When it goes out, negativity inside the cell increases. It increases from -70 millivolt to -87. At this point, the channel is maybe opened, but potassium doesn't go out because negativity inside the cell is so huge that doesn't allow further movement of potassium outside the cell. Right? So, Nernst potential. Now, based on this, we can conclude that if a neurotransmitter open sodium channel, then the membrane potential changes toward Nernst potential of sodium. It's positive. If a neurotransmitter opens potassium channels, then membrane potential changes toward more negative values. Right? And you know that we have a concept called threshold of action potential. Threshold of action potential is usually more positive than the resting state. So the resting membrane potential is -70, the threshold is maybe -50. Right? So, if the change of potential in response to the neurotransmitter is toward positive values, it means potential changes toward threshold. So that's why we call this excitatory. Right? If a neurotransmitter causes opening of other channels that changes the potential downward, so potential goes away from the threshold. So we call this inhibitor. Why? Because it's going away from the threshold, the probability for having action potential is less. When it goes toward the threshold, probability for having action potential is high. Got it?
Yep.
I think it's very simple for you. Okay. The neuron in the resting state. Resting membrane potential is -65. This presynaptic terminal releases a neurotransmitter. We don't know the name. Maybe it's glutamate or acetylcholine. That opens sodium channel. Sodium goes into the cell, right? There is a change from -60 to -41. Negativity is lower, right? So we have more positive value. So is it excitatory or inhibitory?
Excitatory.
Excitatory. How much excitation do we
20.
have?
20.
20 millivolt, right? So from -65 to -45. The difference is plus 20 millivolt. So it's excitatory. So it's happening in a synapse. So it's a synaptic potential. And this synaptic potential is excitatory. So we call this excitatory post-synaptic
Synaptic potential.
potential. excitatory postsynaptic
Okay.
EPSP. So the size of EPSP here is plus 10 millivolts. It's closer to the threshold, so it's excitatory. Right? Now, in this example, a neurotransmitter is released. Let's imagine that it's GABA.
Okay.
It opens chloride channels that causes inward movement of chloride, or opens potassium channels that causes outward movement of potassium. Let's consider this now. Potassium goes out,
Okay.
the cells become more negative.
Negative.
Why?
Because
Nair's potential of potassium is
negative.
negative. Nair's potential of chloride is minus 69. It's 1 millivolt more positive than minus 70 here.
So it's near.
So it's excitatory. No, I think it's inhibitory, sorry. From minus 65,
Yes.
right?
And it's
Okay.
focus.
So
Inhibitor.
both of these causes inhibition. Why? Because they change membrane potential from
Inhibition.
minus 65 to minus
70
IPSP, inhibitory postsynaptic
potential.
potential. So, acetylcholine in the skeletal muscle binds to nicotine receptor, which is a sodium channel, right? So, open sodium channels, sodium goes into the cell, we have EPSP. The same acetylcholine in the heart binds to muscarine receptor, which is a metabotropic receptor that opens potassium channels. Potassium goes out, the cell becomes more negative, so it causes IPSP. So it's inhibitory. So the same neurotransmitter causes EPSP in one place, IPSP in another place. So it's excitatory in one cell, it's inhibitory in another. Got it? Good. So, read this one by yourself, okay?
Can you explain?
Does it have knots?
Yeah, it has no. If you didn't understand, you can ask me later, okay? This is the same thing I mentioned in the first part. Axosomatic, axodendritic, axonic synapses, right? Let me talk about this one. This is more important.
So what inhibitory and excitatory impulse we just depend on the charge of the ion, or the
What depends on charge?
excitatory and
Excitatory
inhibitory.
and inhibitory. If it changes the membrane potential toward positive values, it's excitatory. If it changes the potential toward more values, it's inhibitory. For having this, it may act on different receptors or on different channels. If sodium channel is open, it's excitatory. If potassium channel,
So it depends on the charge of the ion.
charge of the ion, the type of the receptor, the type of the channel, all of that. Okay. So, here we have an axodendritic synapse, right? Not an, several axodendritic synapse. So this one is a synapse, this one another synapse, another synapse, another one. Okay? So, a cell usually receives not only one axon terminal, usually several axon terminals, right? So, another thing. Let's consider this one. Here. This is a terminal which is excitatory. I mean, it releases a neurotransmitter like glutamate, which opens sodium channel. Sodium goes into the cell, resting state is minus 60 millivolts. Now, because of this inward movement of sodium, potential changes to minus 10 millivolts. Do we have EPSP or IPSP?
APS.
EPSP, exactly.
Because-
Why?
Because it's
That's
entered.
more positive than 65.
Yeah, minus 10 is more positive than minus 60. How much is the size of the EPSP here?
Minus
50.
50 millivolts. Okay. I told you before that in a cell, action potential is usually made in the initial segment of the axon. Here,
Yes.
right? So any change of potential in the tip of the dendrite should reach here and change the membrane to the tertiary. Right? So change of the membrane should happen here. But it's happening here now. Right? But it should go through the dendrite to the soma, from the soma to the initial segment. But in this way, you can see that there is decrease in the potential. At first it was minus 10, then
20,
it became minus 20, minus
35,
30, minus
50,
50,
60.
right?
Yeah.
Why is this happening?
Because there are some resistance.
There is some resistance inside the dendrite. One reason,
Diameter.
we have leaking. Resistance depends on the thickness. Leak. So ions come in response to this inward movement of sodium, potassium goes out. Right? So outward movement of potassium nullifies or resists the action of sodium. Right? So, in this movement we have decrease of the potential. This one is called decremental conduction. The decrease in the size of EPSP or IPSP from the tip to the base of the dendrite is called decremental
Conduction.
conduction.
That's a-
Okay, it's happened in dendrite. Why? Dendrites are thin, so there is high resistance for electrical charge. The second one is dendrite is leaky, so when ions come in, some other ions go out, so they leak outside. So that's why we have decremental conduction, right? Another thing, I told you before that in a dendrite we have low density of sodium channels and potassium channels. So usually it's not possible to have transmission of signal through the membrane. So how it's possible to have the transmission? Cytoplasm.
Stream. Cytoplasm.
By cytoplasm. You know that you know from physics that electrical charge can go through a metal wire or through an ionic solution. Right? So we can say that transmission through a membrane is similar to a metal, but transmission through the cytoplasm is similar to ionic solution because we have ionic solution here. It's full of water and ions. Right?
Okay.
So this transmission happens through the cytoplasm, not through the membrane. So that's why we call this electrotonic conduction. So electrotonic conduction means conduction of electrical signal
Passing
through the
from-
cytoplasm of the dendrite.
To initiate the process.
This is subject to decrease. So we call this decremental conduction. Got it?
Yes.
So, another thing, I told you that a synaptic activity here should change the potential here to the tertiary. If we have decremental conduction, then it cannot change the potential to the tertiary. How can we have action potential? Because there is decrease. When it comes here, it's very small. Right? how it can change the potential to the threshold. So the answer is by combination, by summation. Okay, so we have a synapse here. At first, it makes an EPSP of plus 50 millivolts. When it reaches here, only 5 remains. Okay, or only 10 remains. 10 is not enough. But we have another synapse here. It can also make a 5 millivolt change in potential. We have also other synapses here. They can also make a 5 millivolts EPSP. So 5 from this, 5 from this, 5 from this, 5 from other synapses combined together. So they make a very large potential, which is enough for changing the potential to the threshold. Remember, in the soma we don't have leak of the ions for resistance. So there is no decrease in the size of potential in the soma. Okay? So everything that reaches here remains here. Okay? So we have summation of signals.
So we have leukopoint exons.
In the dendrites only. In axons we don't have. Okay?
Sir, synapses take information from axon to dendrite, not from dendrites to axon. So why we have more excitatory and inhibitory neurotransmitters like near the tip of dendrite?
I didn't get your question.
Synapses take information from axon to dendrite.
From axon of the presynaptic cell to dendrite of the postsynaptic cell.
So why we have more inhibitory and excitatory neurotransmitter like near the tip of dendrite?
So the tip of the dendrite here, you mean this. Okay? So it receives axon terminal from the presynaptic cell. So this one is from presynaptic cell. And this is from the postsynaptic.
Postsynaptic.
So it releases, it receives neurotransmitters from the presynaptic axon terminal. Got it?
Yeah.
Okay, so let's see this one now. Actually, you're out of time. So let me talk about this later. Do you want?
Yes, okay.
It's better. So in the next session tomorrow at 10,
Yes.
first I will talk about this and then I will continue the next topic. Okay? But